the Kenya Renal Association for inviting me to your 2021 meeting. And I would like to acknowledge the many contributors to this presentation on the genetic predisposition to chronic kidney disease in Africans and the role of apolipoprotein L1 gene variants. I would particularly like to acknowledge Professor Jacob Kleins-Ruhl, who was a key contributor to our studies, who unfortunately died last year. And I'd also like to acknowledge the enormous contribution to kidney disease of Professor Anthony Were, who also died earlier, and two great nephrologists who have died within the last year and a bit. And I pray that God grants their souls rest in eternal peace. I'm presenting this on behalf of the H3 Africa Kidney Disease Research Network investigators. We currently have completed or are ongoing 4 studies, the kidney disease study, and I'll show you data from that. The cohort study, I'll show you data from that. We've got ongoing studies, collaborative centers, and the COVID-19 studies, and I won't be able to show you data from that. I'd like to acknowledge my co-principal investigators, as Professor Akinlolu Ojo from Kansas University Medical Center and Professor Salako from College of Medicine, Ibadan University. Now, the prevalence of chronic kidney disease in Africans is of the order of 14%. And also Africans succumb to end-stage renal disease some 20 to 30 years earlier than Europeans. Renal replacement therapy is either unavailable or unaffordable, making the development in many countries of end-stage renal disease a death sentence for many. And chronic kidney disease extracts an enormous toll in premature mortality and cardiovascular comorbidities. Now, we know that African Americans have a 3.7 times higher age-adjusted risk of end-stage renal disease as compared with European Americans. And studies by admixture mixed disequilibrium technique showed that polymorphisms in apolipoprotein L1 were associated with end-stage renal failure. And this technique of mapping by admixture linked disequilibrium basically tries to map the risk of kidney disease in Africans to either European or African genes. And in so doing, they were able to map that risk to apolipoprotein L1 gene variants, which were then shown to be strippinolytic. And these were groups in 2010 from Harvard, Martin Pollack's group, and from Israel, Karl Skorecki's group, showed that these mutations were associated with end-stage kidney disease. And so the apolipoprotein L1 gene variants, on the one hand, kill trypanosoma brucei and so the normal apolipoprotein L1 has no effect on trypanosomes, but the G1 and G2 variants will lyse trypanosomes. And some 10,000 years ago, when trypanosomiasis was common, having these variants provided protection against sleeping sickness and therefore death. And by the law of unintended consequences, these same variants that protect from trypanosoma lead to kidney disease. And if I show this graphically, so individuals who don't have these variants get trypanosomiasis, sleeping sickness, and often succumb, whereas individuals who have a variant, and there are 2 variants, G1, which was a missense mutation in the apolipoprotein L1 gene, and G2 is a deletion. So individuals who had one risk allele were protected against sleeping sickness, a little bit like individuals with sickle cell trait are protected against malaria. Individuals with 2 risk alleles, again, were protected against trypanosomiasis, but unfortunately developed kidney failure. And if you look at the distribution of these apolipoprotein L1 variants, that's the G1 and G2, what's really quite striking is that although Trypanosoma brucei rhodesiense is common in Central Eastern Africa, the prevalence of the APOL1G variants is much higher, of the order of 35 to 50% in West Africa, particularly Ghana and Nigeria. And the G1 variants, of course, provide protection against rhodesiense. And the G2 variants, again, were much more common in Ghana and Nigeria, perhaps less so than in East Africa. And the reason for the protection, the G1 and G2, given why they rose to such high levels in West Africa as compared to East and Central Africa, is still not known. So the apolipoprotein L1 gene is associated on chromosome 22. Apolipoprotein L1 is produced in the liver, circulates with high density lipoprotein L3. And the two risk variants that I've mentioned, G1 and G2, and explain much of the excess risk of chronic kidney disease in African Americans, and increasingly we're observing in Africans, and is the strongest risk factor for chronic kidney disease. So in terms of trypanolysis, ApoL1 enters the trypanosome in conjunction with high-density lipoprotein, separates from that, and the ApoL1 inserts itself into lysosomal membrane and lyses the lysosomes. Quite high cause of kidney disease is not known. It may alter the structure of podocytes. It may again cause lysis of podocytes. It enters the mitochondria and may again cause lysis and damage, and also the endoplasmic reticulum. And the mechanism of apolipoprotein L1 associated kidney disease is a source of considerable research at the moment. If you look at ApoL1 nephropathy spectrum, there are some diseases that are significantly associated with apolipoprotein L1 with odds ratios of upwards of 20 or higher. And these are HIV-associated nephropathy, focal segmental glomerulosclerosis, and hypertension end-stage renal disease. And by contrast, IgA nephropathy and diabetic kidney disease are not significantly associated with apolipoprotein L1 variants, and the reason for these differences are not known. And it's obvious that not all North African Americans or Africans with ApoL1 variants develop, high-risk ApoL1 variants develop chronic kidney disease. So there must be second hits that are either gene or gene environment. And one such environmental factor is interferon. So some African Americans treated with interferon have developed a focal segmental glomerulosclerosis and a collapsing glomerulopathy. And one of the strongest associations for ApoL1 associated kidney disease is HIV infection, which of course induces an interferon production. So the ApoL1 has been the focus of our studies over the last 8 to 9 years. And the H Africa Kidney Disease Research Network have phenotyped 4,000 CKD cases, 4,000 controls, and then conducted genetic and translational research projects, and also had a huge emphasis on training of clinical research personnel and genomics investigators. And just to show the three main studies, there's a Kidney Disease Research Network where we recruited a large number of cases and controls, and of those who conducted genome-wide association study, actually now in some 5,000 cases. There's a cohort study that actually sought to target biopsy-proven glomerulonephritis as well as chronic kidney disease. And we've got the more recent collaborative center study that's focusing on childhood nephrotic syndrome, sickle cell disease, and sickle cell trait. And the centers have actually all been in West Africa, and that's something we would like to extend to East, Central, Southern Africa, so 11 centers in Nigeria, and 2 in Ghana. And we've looked at genome-wide association, prioritized pathways, look for prototype-specific genes, develop gene-gene and gene-environmental interactions, and look at population variation across sub-Saharan Africa. And we actually defined chronic kidney disease along conventional lines with an albumin creatinine ratio greater than 3 millimole per liter, estimated GFR less than 60. Define some CKD cases as just an eGFR less than 60, and then some as both an eGFR less than 60 in patients with biopsy-proven kidney disease. And by definition, all our controls had an eGFR greater than 60 and an albumin creatinine ratio less than 3 milligram per millimole. And we genotyped the APOL1 gene variant G1 and G2 using custom Tachman assays with a high degree of stringency. And this shows our initial APOL1 analysis is not complete yet. The numbers are going to be greater. But for this presentation, I thought I'll show this to you. So we've analyzed data on 5,531 cases and controls made up of cases and controls. And there was good separation between the CKD cases and the controls, so that by definition, almost the CKD cases had a higher blood pressure, were more likely to be hypertensive, had a lower eGFR 52 to 99, and by definition were the only ones that had albuminuria. Now, when we look at the association of APOL1 gene variants and chronic kidney disease, this showed you is a recessive model. So we compared 2 APOL1 risk variants against 1 or 0. And as you can see, in the adjusted association was 1.29 odds ratio with a range of 1.14 to 1.45. So this was highly significant for 2 risk variants versus 1 or 0. This was all chronic kidney disease. If we look at chronic kidney disease with a low GFR, no albuminuria, again, the adjusted odds ratio was significantly higher at 1.45, range 1.23 to 1.7. And if we looked at patients with biopsy-proven glomerular disease, childhood nephrotic syndrome, again, the odds ratios were high at 1.49. Odds ratio was high at 1.49. One of the really intriguing things that we found was that within Ghana, a CANS ethnic group had 2 APOL1 risk variants in 37% of the population. And by contrast, Evers and GARS had much lower proportion of risk variants of 17 and 90%. We found the same sort of pattern in Nigeria, where 42% of Igbos had 2 APOL1 renal risk variants. It was lower at 23% in Yorubas and much lower at 12.1% in Hausa Fulanese. And the reason for this aren't clear, but must reflect migration patterns and then for the exposure to trypanosomiasis over the last 10,000 years. So to summarize our findings, 28% of our entire study population, Ghana and Nigeria, got 2 APOL1 high-risk variants. The G1 haplotype was more frequent at 36% than the G2 haplotype at 13.5%. And individuals with chronic kidney disease had a prevalence of APOL1 high-risk variants of 30.6 against 25.9, the control population. And as I've shown, the association with chronic kidney disease of high-risk carriers was 1.29 times higher as compared to low-risk carriers. I haven't shown you the data, but the APOL1 effect on chronic kidney disease appeared to be greater in men than in women. We actually didn't find a differential effect of sickle cell disease, although the numbers are small, and on diabetes mellitus in keeping with other studies. And we found an ethnic disparity in APOL1 high-risk genotype that we're yet to explain. If I move on to the cohort study, we so far enrolled some 3,337 cases, of whom 706 had biopsy, and we're following these patients up over 4 years. And all the renal biopsies were done by Jeff Hodgins at Michigan University. And although the samples had to travel all the way there, got very good definition. So somebody was membranous and the chain linking, good immunofluorescence, and also excellent electron microscopy. And we also, looking at disease clinical pathological category in 600 cases so far, focused segmental glomerulosclerosis, these cases were all in adults, was much higher, 35%, minimal change, 17%, and membranous in 9.7%. And this is one of the largest biopsy series in sub-Saharan Africa. And Jeff and his team are creating a pathology infrastructure with digital pathology imaging that is then placed on the internet so that we can then learn from it and also do analyses of the different diseases against their genomics. We also sent off renal biopsies to look for signatures of chemokines and cytokines. And this shows one such heat map across 52 biopsies, showing expression of, in this case, TNF, that was much higher in some biopsies than in others. And when we look at it, we found that patients with focused segmental glomerulosclerosis were much more likely to have TNF than, for example, hypertensive nephropathy. So this begins to give us a handle on the pathophysiologic mechanisms associated, if not leading, to these diseases. Again, the one thing that the H3 Africa Kidney Disease Research Network has been very keen on is community engagement, explaining our research to our communities that we work in, either at gatherings, meetings, faith-based, work-based meetings, to try and ensure that people understood at patient gatherings, try and ensure that people understood what we're trying to do. And this is myself, my colleague, Dr. Safo, meeting with community elders, and would meet with the community elders and then with the community, hand out information, and then go back later to do the research. And we're now developing mechanisms of feeding back the genetic and other information that we've found in our studies to our patients. And we've also contributed to the development of an ethical framework for genomic research in Africa involving our people, so that no longer would people parachute in, take blood, isolate DNA, and do genomic studies out of Africa. So we're trying to make sure that our research is embedded within Africa. Just very briefly to mention, because I know that Kenya does a lot of kidney transplant, that the risk, if there's any, of the APOL1 variants resides with the donor. So if there's a high-risk donor, they're perhaps more likely to pass on the disease to the recipient. And if the recipient is high risk, then that is not associated with an increased risk. And there is now ongoing study, the Apollo study in the US, so we're not sure how real that risk is. So we've currently got an ongoing cohort study that I mentioned, got the collaborative center study, childhood nephrotic syndrome, translational studies in sickle cell disease, and ongoing studies in COVID-19 infection. We've also got collaborations within and without the H3 Africa Consortium, and most of these are ongoing. Dr. Safo, who's one of our at the University of Ghana Medical Center, is currently doing a study on apolipoprotein L1 and preeclampsia, as indeed a group from Tennessee. Dr. Safo has done a study looking at isotopic and estimated GFR in West Africans. We are collaborating with the cardiovascular disease working group. Professor Labisi is generating mature podocytes from inducible pluripotent cells derived from our patient's blood. And then with tissues at Boston, we're looking at dietary sodium, potassium, and ApoL1 nephropathy. We published a paper on environmental pollutants and chronic kidney disease in children. Also a paper on pathogenic urinary tract viruses, polyoma, and ApoL1 associated kidney disease, and on the genetics of breast cancer. Currently ongoing study with University of Alabama on the REGARD study and the University of Utrecht on sequencing of genes involved in congenital kidney disease in young Africans. So since end-stage renal failure without renal replacement therapy leads to death within days to weeks, the identification of gene variants associated with kidney disease progression phenotype is of existential significance to millions of Africans who already have chronic kidney disease and many more at increased risk amongst the nearly 1 billion living in sub-Saharan Africa. And this is a quote by Professor Akinlolojo, one of my co-investigators. It just remains to thank the National NIDDK, the National Human Genome Research Institute, my collaborators, and this was at one of our consortium meetings in Ibadan. And it's not all been hard work. We had a consortium meeting at Victoria Falls, and these are my collaborators enjoying myself. And of course, for H3 Africa, for allowing us to do this genomic research in Africa. Thank you for your attention.
Thank you, Professor Adu, for that very interesting presentation on H3 gene, APOL1 gene in Africa. Our next speaker is Dr. John Otieno. We want to apologize for a change in topics. We were supposed to have Milton Obilo talking to us on pleiotropic effects of SGL2 inhibitors. Unfortunately, he had to leave suddenly because he was informed that his mom passed away. So we send our condolences to his family, and we pray that the Lord Almighty rest her soul in eternal peace. So we come back to Dr. John Otieno. Can you please come to the podium?